Hello, welcome to Beyond the Reference, Genomics for Amphibian Research and Conservation. I'm Dr. Tiffany Kopp from the University of Melbourne. I'd also like to welcome, today we'll feature 3 excellent talks from Amphibian Genomics Consortium members, but first I'd like to take a moment to tell you more about the AGC. The AGC was launched in early 2023 to unite amphibian researchers across the globe and increase genomic research on this fascinating and highly threatened group of vertebrates. Similar to other genomics consortia, the AGC endeavors to generate amphibian reference genomes, but we are also focused on moving beyond reference genomes to better understand amphibian ecology, evolution, and to solve their conservation challenges. Additionally, we aim to support the sequencing of large, complex genomes, such as those of many salamanders, which have been largely neglected by most sequencing initiatives. The AGC is an inclusive organization. We welcome members from the conservation community and researchers outside the field of genomics. We encourage participation from members across time zones by having meetings at alternating times and use communication channels to further facilitate communication between geographically dispersed members. Our main aims include funding genome sequencing, improving genome annotation, providing computational resources and workshops, generating technical resources and best practice guidelines for working with omics data, and supporting students and early career researchers. The AGC's leadership consists of 3 directors and a 10-member board. The board was selected to ensure gender equity, diversity of backgrounds and career stages, and geographic representation. This map shows the geographic locations of our board and directors. The AGC now has more than 350 members from 45 countries. This bar plot shows the number of members by country, and the map shows countries scaled by members. Although our membership is geographically diverse, there is still a lot of geographic disparity. We plan to increase engagement efforts in the future to increase membership from these countries, many of which are regions of high amphibian diversity and/or endemism, such as South America and Southeast Asia. The rate of amphibian genome sequencing has lagged behind that of other vertebrates, with only 111 of over 8,600 described species sequenced. This is compared to 129 reptiles and more than 700 each of bird and mammal species. Launched at an opportune time, the AGC aims to close this gap by supporting and accelerating amphibian genomic research. This work is essential for advancing amphibian conservation and understanding their evolutionary history, topics we'll explore in today's presentations. One of the AGC's next steps will be to fund sequencing for 48 priority species to fill key gaps in the amphibian phylogeny I showed you earlier. Check out our new BMC genomic paper for the full list. That's all for me for now. Before we move on to our other presentations, here's the QR for the AGC website if you'd like to join. Anyone is welcome and membership is free.
I'd like to start by acknowledging the Wallagaloo and Wiradjuri people, the traditional custodians of the land on which the crocodile frog are native, and acknowledge the Wurundjeri people on whose land this work was conducted, and I pay my respects to their elders, past and present. So hey everyone, I'm Mickey, and I'm right at the end of my PhD. So I'm going to give an overview of my research, which has been working towards using selective breeding as a method to combat chytrid susceptibility in the southern crocodile frog. So traditionally known as Gaiac, the southern crocodile frog is a small, range-restricted alpine species that is endemic to these sphagnum bogs in Mount Kosciuszko National Park in Australia. And back in the mid-1900s, there used to be a pretty prolific species in the park. Legend even has it that people used to collect them off the side of the road to sell as fishing bait. But in the 1970s, the amphibian chytrid fungus was introduced to Australia, and so began their decline. Now, unlike some species, which were completely wiped out within a couple of years of the chytrid fungus being introduced, the corroboree frog's decline was comparatively slow. But by the time they were listed as critically endangered in 2004, they were at imminent risk of extinction. So to try and mitigate this, animals were collected from the remaining wild source populations and brought into captivity to establish captive breeding and assurance colonies. And these have been very successful. However, all attempts to re-establish these frogs back in the wild have failed due to the presence of chytrid in the environment and reservoir hosts. So currently, their only survival strategy is captive management. But the captive management of this species is being done incredibly well. Across 3 institutes, there are approximately 600 founding frogs, which were collected as eggs from the wild between 1997 and 2012. And these founders are from 15 historical geographic populations, which have indicated here on the map. And a study which looked at microsatellites showed that the genetic diversity within the founders was actually pretty high. However, this is now a closed population, with only 25 recorded males calling in the wild this year, all at reintroduction sites. The entire genetic diversity of this species is now held in captivity. Now, these ARC populations are effective in buying a species time while their solution to their threatening process can be sought. But with an aging founding population and no solution to the chytrid problem in sight, this species is either bound for extinction or will remain in a closed captive population. That is, unless we try something different. So that's exactly what my research aimed to do. I wanted to see if we could selectively breed corroboree frogs that are innately more resistant to the chytrid fungus. Now, this isn't an unreasonable approach. Breeding animals for specific traits has been occurring for thousands of years and been very successful. Think cows for increased milk production, chickens for eggs, fish for bacterial resistance, or shrimp for viral diseases. It's just, this has never been tried for a conservation purpose before. But given that we have the corroboree frog's entire remaining genetic diversity in captivity and their breeding is intensively managed, this species presents a unique opportunity to test the efficacy of selective breeding to improve conservation outcomes. Because if this was successful, it would provide a long-term and sustainable solution to establishing a population of corroboree frogs in the wild in the presence of the chytrid fungus. But to do this, we need 2 things. We need the frogs' phenotypes and we need their genotypes, which we can then use to make genomic predictions to inform selective breeding strategies. Now, this seems pretty straightforward, except we were starting from the start with no existing phenotype or genotype information for the species. We had to collect and resource these. So I collected our phenotype and genotype data through experimental chytrid exposures. So using just under 1,000 captive bred juvenile F1 southern corroboree frogs, I performed 3 replicate 14-week exposure experiments. And our aim here was to identify phenotypic variation in response to disease. So I'm going to take you through the phenotypes and genotypes and then how I use these to make genomic predictions for this species. So firstly, our phenotypes. So the 2 main traits we were measuring here were survival and infection loads. And as you can see, we saw variation across the 3 experiments in the number of frogs which survived and the number of frogs which were infected. Now, due to this variation, we analysed each experiment separately and then used a random effects meta-analysis to combine the results of the 3 experiments. So looking at these traits a little closer, for survival, there were significant differences between the 3 experiments. However, the only factor which significantly impacted their survival time was the zoo in which the frogs were bred at, with frogs from one zoo having shorter survival times compared to the others. Now, when we look at infection loads, and these here are our linear transform infection loads over the course of the experiment, the only significant factor was the body condition of the frog. With smaller frogs not only more likely to become infected, but when they were infected, their rate of increase in infection load was faster. But what I found most interesting was that we had around a third of our frogs never test positive. That is, they were exposed to chytrid and tested negative for 14 weeks. And for a critically endangered, highly susceptible species such as the corroboree frog, this was surprising. But it actually replicated the disease dynamic that we saw in the wild before their populations crashed. So now we have the phenotype information we need. We need our genotypes. Now, as the goal of our project was to identify targets for selective breeding, we chose to use a SNP array for our genotyping. And an array seemed like the best approach for this for a few reasons. Firstly, the corroboree frog genome is very large at 8.8 gigabases and has high repetitive content. We also had a very large starting initial data set of 1,000 frogs. And as we were investigating the potential of selective breeding, we wanted a method that was not only repeatable across samples, but also across multiple generations. So we designed and manufactured a custom 50K SNP array for the southern corroboree frog. And the result, the first custom SNP array for any amphibian. So this southern corroboree frog array consists of 48,000 markers and has a density of around 4.6 SNPs per megabase, which I've shown here in the heat plot. And the results of running the array were incredibly positive. 95% of my samples passed quality control, and 83% of the SNPs converted polymorphic genotypes. So now we have the genotypes of those 971 F1 frogs. But before we can use them to make our predictions, we first need to check a few things. Firstly, we wanted to see if there was any population structuring in case some animals or families needed to be treated as genetic outliers. And we also wanted to look at patterns of linkage disequilibrium across the genome, as this can determine statistical power and accuracies in genomic studies. So to look at population structuring, we used a PCA which showed that axis 1 and 2 explained approximately 15% of the genetic variation within these animals. DAPCs and structure plots also indicated that there was no major population substructuring in the F1 animals. Now, this is actually what we would expect to see, as the breeding design of these frogs is based on a maximum avoidance of inbreeding groups, which crosses males and females from different subpopulations. So moving forward, we're treating them as one population. And for our linkage disequilibrium analysis, we looked at SNPs across the 12 chromosomes, and these showed an average LD of 0.6 with an average intermarker distance of 216 kilobases. And as you can see here in the heat plot, the LD decay, which is shown in orange, is very slow, not reaching background levels until around 900 kb. So this indicates that we would require approximately 11,000 SNPs for accurate genomic predictions. So this was pretty good news, as we have triple the number of SNPs on the array, making it a very powerful tool for genomic selection in this species. So now we have the genotypes of the frogs, and we know we have sufficient SNPs to make accurate predictions. We can put this data together with the previously mentioned phenotypes to make our predictions. So the first thing I did was to calculate the heritability of traits. Now, this is important because if we want to implement a breeding program, then the trait we select for must be heritable. If it's not, then breeding will not cause any additive change to that trait. In total, I measured several life history and morphometric traits, but for simplicity, I'm just going to present the results of survival, which was measured in days, as this is what we were really interested in: survival when exposed to the amphibian chytrid fungus and how we could improve this in the corroboree frog. And thankfully, survival in this species is heritable, with a heritability of around 20%. Now, while this is what we would term low heritability, it is still heritable. It just means that progress will not be as fast as we would have liked it to have been. So having established that survival is a heritable trait, we next wanted to determine the genetic architecture of this trait. So we conducted a genome-wide association analysis. Now, GWASs allow you to identify genomic variants that are statistically associated with a measured phenotype. And what you're hoping for is there'll be one gene that has a major effect on your phenotype, and you get this nice big peak in your plot. But disappointingly, but not surprising, was that there was no SNP of major effect on survival. What we see here instead is many SNPs with minor effects. So the trait is polygenic, meaning survival when exposed to chytrid is controlled by many genes. And this makes sense because if there was a gene of major effect contributing to their survival, it's likely that it would have become fixed in the population, and corroboree frogs wouldn't be in the dire situation that they are in now. But because our trait is a polygenic heritable trait, selective breeding is a viable method to make immediate improvements in survival in this species. So, to test this, I modeled different selective breeding strategies. Now, these models can take into account the number of animals or matings, the relatedness between individuals, and the goal of your selection. So the three main selection goals I wanted to look at was to minimize inbreeding in these animals, to maximize genetic gains, which here is our survival, and an optimized contribution approach, which tries to balance inbreeding with genetic gains. And I align these models as realistically as possible with the biology of this species, only allowing females to contribute once per breeding season, whereas males could contribute up to 3 times. And I use 200 frogs as this is the approximate number of F1 animals that we have in each breeding colony. So if our goal here is to minimize inbreeding, then we use a model which ignores the animal's estimated breeding values and just aims to minimize relatedness between individuals. And what you can see is that we're actually able to increase heterozygosity in the population with a negative inbreeding value, but there is very low genetic gain, with an average increase of 1.1 mean days survived in the next generation. And so we see the opposite effect when our aim is to maximize genetic gains, as this ignores the relatedness between individuals and only uses our top performing animals. So while we have much higher genetic gains with an increase of 4.9 mean days survived in the next generation, we also have significantly increased inbreeding. And typically, the goal in breeding programs is to keep inbreeding below 1% per generation. So this is not a good model. But it brings us to our optimized contribution approach, which tries to balance inbreeding with genetic gains. And the takeaway from this is that we can achieve a level of genetic gains not too dissimilar from our maximum gains model, while also balancing inbreeding below that target of 1%. So what does this realistically mean for the species? Well, the current generation, our F1 frogs, has an average survival time of 77 days when exposed to chytrid in a laboratory setting. So if we were to apply an optimized selection approach to their breeding, then we can increase the mean survival time to 82 days in the next generation. So in theory, with each generation, this increasing the frog's ability to survive infection. So this work, well, it provides us with the first real evidence that applying a selective breeding approach to the captive southern corroboree frog population could result in tangible outcomes to improve their conservation. The first real hope this species has had in a long time. However, this project still has a long way to go. My PhD was just the beginning, but my work shows us that selective breeding is a viable method. So the next steps are to cross-validate our results using different prediction models. We then need to determine if our predictions actually result in realized gains. And additionally, we need to check that the laboratory results will translate into the field. And finally, for this talk, but not for the project, is that we need to continue monitoring and assessing traits to ensure that there's no negative outcomes of selection. So with that, I'd really like to thank all the people and organisations directly and indirectly involved in this project, as well as you all here for listening. So thank you.
Hello, everyone. My name is Ying Chen. Today, I will share you my PhD work on the chromosome-level genome assembly of Western chorus frogs and insights on the chromosome evolution, rebel genes, and the new sex-linked region in anurans. So I will start from a brief introduction on the arborano frogs, or the tree frogs. Tree frogs are adapted to live in trees, bushes, or other vegetations. They have those adhesive toe pads that help them climb trees and leaves. The entire clade originated in South America in late Cretaceous or early Cenozoic and have diverged into over 900 species and 3 families. Highlighted here is the largest family with 7 subfamilies and 49 genera, and most species are distributed in the Middle and South America. 2 ancestral clades reached North America. One is the ancestor of hylae and bryophytes. Some of the species even reached Eurasia. The other clade is the ancestor of subfamily Acrocinae. The species are now endemic to North America. Currently, only 2 genomes are available for the entire Operano frog clade. So we are interested in the species Western chorus frog. It's a small tree frog species in eastern United States and Canada. They breed in the early spring in temporary ponds and are very cryptic in the rest of year, hiding in terrestrial lands. It's a species of conservation concern. Populations are declining in many areas, and some of them are listed as threatened in Ontario and Quebec, Canada. They also have complex evolutionary history with mitonuclear discordance patterns found. And this suggests some historical hybridization with non-sister species. So to facilitate conservation and to investigate the evolutionary relationships, we collected a male individual from Canada and assembled a reference genome. We used the latest PacBio review system to generate the hi-fi line reads and the Dovetail Genomics Hi-C kit to generate a contact map data. We assemble and phase 2 haplotypes in hyphaism, scaffold the contigs in YAS, examine possible contamination in block toolkit, and remove mitochondrial contigs by blasting the assembled mitochondrial genome assembly against the nuclear assembly. And we use the TreeBuild pipeline and the rapid curation pipeline to generate necessary Hi-C files for manual curation in pretext view. So the final assembly was 4 gigabytes in size with over 1,000 scaffolds. The scaffold M50 is 441 million base pairs. The mercury tamer analysis showed good phasing in the final haplotype assembly. The Hi-C contact map showed 12 chromosome level scaffolds here. The Busco analysis showed the genome is 90% completed, and the per base consensus accuracy is 61. The K-mer completeness is over 98% for 2 haplotypes. So overall, our genome has high contiguity and completeness. We then identify the repeats in the genome using the Earl Grey transposal element pipeline, and then annotate the genome in Breaker 3 pipeline using RNA-seq data from 6 tissue samples, including liver, eye, kidney, and testes, as well as the ortho-DB vertebrate protein data. So overall, we predicted over 22,000 protein coding genes, and the Busco completeness is 94.4%, among the highest of the available frog annotations in NCBI database. The OMARG quality assessment, which is similar to Busco but has much larger database, also showed very high completeness and no evidence of contamination. So overall, both our genome and annotations are of high quality. So using our newly generated genome, we first examined the synchrony with additional 22 frog genomes from NCBI database. We labeled the chromosome numbers for each species at the end. Each color here represent a candidate ancestral chromosomal block, also labeled as letters on the top. And this is modified from Bredesen et al. 2024 paper. So overall, the chromosome synchrony and collinearity is highly conserved throughout over 220 million years of evolution. There are 2 important notes I want to mention. The most ancient lineage coastal tailed frog here has many small chromosomes or microchromosomes, and those chromosomes broke and then fused into larger chromosomes and then become largely stable across the 200 million years of evolution. What's unique for the tree frog clade family here, the Hylidae family, is this chromosome element E here showing us the coral color that broke into 2 and then fuse with 2 chromosomes. This fission and fusion events is pretty rare compared to other chromosome blocks that experience fusion and fusion largely intact. So with such conservatism in synteny, we ask whether there are any rebel genes that do not follow this general pattern and are thus potentially important for evolution. So we use this network-based microsynteny analysis here. So each node here is a single gene for each species. And if this gene is syntenic across all species, they will be connected by edges in the network. And if the gene is only syntenic in a few species, then they will become separate networks for this gene. So we use this analysis we did this analysis on 29 frog species, and indeed, most of the Busco genes are one copy and syntenic across all species. But here, we see some of the genes are almost duplicated in almost all frogs. And though some of them are indeed relate to some important functions for frog evolution. I will make one example here. As we all know, many organisms use vocal communications, including frogs, geckos, birds, bats, and humans. Frogs and toads are among the most ancient lineage that produce calls to attract females for mating, and the temporal attributes of the calls, such as pulse rate and pulse duration, serve as pre-mating isolation mechanisms and are therefore important speciation traits. So what's essential here is the neuron circuit architectures that support call production. The electrophysiological experiments show that interval counting neurons that bandpass selectivity for pulse rate and interval counting, and the tuning of the neurons are mediated by the excitatory inputs via inotropic glutamate receptors and inhibition via the GABA receptors. The upregulation of one or more genes that encode these GABA receptors may increase the relative amplitude of inhibition, and as a consequence, increase the number of pulses. So we found this GABA receptor subunit alpha-3 gene that has 3 or 4 copies in almost all frog genomes, which we hypothesize might underlie neuron development and calling behaviors. So back to the rebel genes, among 7% genes here that has 2 or more, that 7% has 2 networks, and some of the genes are listed here that are transposed to a different chromosome in the modern frogs, Neobacteria clade. And so future studies can investigate those genes to see whether they play a critical role in the Neobacteria frog divergence and evolution. We still have very limited knowledge in terms of the function of those genes, so we listed the genes here for future studies. So the next thing we are interested in is the anurin sex chromosome. Around 75% of the anurins have homomorphic sex chromosome, making it very challenging to study their sex evolution. And unlike mammals and birds, they have smaller Y and W. For those species with heteromorphic sex chromosomes, anurin has bigger Y and W. So what we also found is that the sex chromosome are not a single homologous chromosome for all species. In clades like Rhinidae frogs and Pipidae frogs, many species use different chromosome pairs as their sex chromosome. But for Halidae frogs and Bufonidae frogs, their sex chromosome seems to be largely conserved for this one single chromosome pair. And so we ask whether that's also true for Western coarse frogs, which is in the Hylidae family. So what we did is we used this, we generate dd-rat sequencing data for 10 female coarse frogs and 50 male coarse frogs. We used the program RatSex to investigate the sex-linked markers. And indeed, we found about 10 markers that are only found in males, and no females has those markers. And when we map those markers onto the chromosome level reference genome, they are all mapped to the same region in the chromosome 1. So based on the analysis, chorus frog also has XY system showing the male specific markers, and they are all, and the sex chromosome are indeed conserved and highlighted as this same chromosome pair. So next, we look at the sex-linked region in this chromosome 1. So we use the mini-map2 to align two chromosome level haplotype assemblies, and it found no synchrony in this region, showing in dot plot. And so in this linear syntactic view, we can see that the Y chromosome, or the paternal haplotype, has a much larger size than the maternal or X chromosome. So we think there's an indel structural variance in this region, and it's not caused by assembly incompleteness, because the gaps are found in those two regions. So in addition, we found this gene in the upstream of this sex-linked region. That's the CNOT6L gene. And this gene is found to be essential for female mice reproductive endocrine regulation, and knocking out this gene causes female mice infertility. And so we think that this gene might play some role in chorus frog sex development. And I want to quickly mention that the structural variance underlying sex determination is also found in different frogs. They also use the same chromosome pair. It's also an XY system, and they also find a larger region in the Y chromosome here than the X chromosome. And this non-coding RNA is Y-specific. That's underlying the sex development and regulate the downstream gene BOLD1L. So we think that structural variants might be important in frog sex determination. So to summarize, we assembled and annotated a new genome in the subfamily Acrocinae. We found conserved synteny in this genome along with other frog genomes. We identified some rebellable genes that might be important for frog evolution. And we identified the sex chromosome and sex-linked region in the chorus frog. So finally, I want to thank all the funding sources and all the people who helped with my PhD work. Thank you. I would love to take any questions if you have.
I'd like to start by acknowledging the Wallagaloo and Wiradjuri people, the traditional custodians of the land on which the corroboree frog are native, and acknowledge the Wurundjeri people on whose land this work was conducted, and I pay my respects to their elders, past and present. So hello everyone, I'm Mickey, and I'm right at the end of my PhD. So I'm going to give an overview of my research, which has been working towards using selective breeding as a method to combat chytrid susceptibility in the southern corroboree frog. So traditionally known as Gaiac, the southern corroboree frog is a small, range-restricted alpine species that is endemic to these sphagnum bogs in Mount Kosciuszko National Park in Australia. And back in the mid-1900s, there used to be a pretty prolific species in the park. Legend even has it that people used to collect them off the side of the road to sell as fishing bait. But in the 1970s, the amphibian chytrid fungus was introduced to Australia, and so began their decline. Now, unlike some species, which were completely wiped out within a couple of years of the chytrid fungus being introduced, the corroboree frog's decline was comparatively slow. But by the time they were listed as critically endangered in 2004, they were at imminent risk of extinction. So to try and mitigate this, animals were collected from the remaining wild source populations and brought into captivity to establish captive breeding and assurance colonies. And these have been very successful. However, all attempts to re-establish these frogs back in the wild have failed due to the presence of chytrid in the environment and reservoir hosts. So currently, their only survival strategy is captive management. But the captive management of this species is being done incredibly well. Across 3 institutes, there are approximately 600 founding frogs, which were collected as eggs from the wild between 1997 and 2012. And these founders are from 15 historical geographic populations, which are indicated here on the map. And a study which looked at microsatellites showed that the genetic diversity within the founders was actually pretty high. However, this is now a closed population, with only 25 recorded males calling in the wild this year, all at reintroduction sites. The entire genetic diversity of this species is now held in captivity. Now, these ARC populations are effective in buying a species time while their solution to their threatening process can be sought. But with an aging founding population and no solution to the chytrid problem inside.
Hi, my name is Hayley and I'm a PhD student at Oxford Brookes University. And today I'm going to be talking about the first half of my PhD project on novel endogenous retroviruses in Anura. So endogenous retroviruses, or ERVs, are viral sequences found in vertebrate genomes which represent remnants of past retroviral infection. Retroviruses become endogenized when they integrate into the germline of a host, and this is subsequently passed to offspring and either lost through genetic drift, or it can become fixed in the genome of a host species or a population. Although the majority of ERVs are lost through genetic drift, they can make up a substantial portion of vertebrate genomes. For example, in humans and mice, they make up around 10%. Within the genome, they provide a source of coding and regulatory sequences which can be co-opted by the host. An important example of this is syncytin genes. Syncytin genes are derived from the envelope or ENV gene of retroviruses and have vital roles in placental development. They've been co-opted multiple times throughout placental mammal evolution. Although there are some examples of endogenous retroviruses which have infectious counterparts which are still circulating, such as in koalas, the majority of ERVs represent remnants of past infections, and many appear as ancient insertions without closely related exogenous forms. By investigating ERVs, we can understand the history of retroviral infections, as well as the host-virus interactions, such as cross-species transmission. For example, a study of ERVs in multiple vertebrate groups found that ERVs with shared common ancestry. So we can see here between mammals and turtles, for example, or in birds and snakes. And this indicates transmission events between these vertebrate groups. So ERVs are formed of 3 genes, GAG, POL, and M, and in the host genomes, they are flanked by long terminal repeats, or LTRs. ERVs are distributed in the family Retroviridae, and this classification of 7 genera, as well as some other groups which don't fit this classification, is determined by the POL gene. And this also shows us the distribution in vertebrate groups. However, there are some processes of recombination which can occur, such as M swapping, which can result in a different gene structure of retroviruses, such as where an M can be gained from a different retroviral group. And when we look at these recombination events, rather than just the POL, we can see that there is a wider distribution. For example, alpha retrovirus, which is host-specific to birds. When we look at the M gene as well, we can see that there is further distribution to other vertebrate groups. So although that we can see that these retroviruses are distributed across various vertebrate groups in this figure, there is the majority of ERV research has focused on mammals. An interesting comparison to mammals is the Anuran group of amphibians, and this is an order of amphibians made up of frog and toad species. They have a similar number of extant species compared to mammals, as well as comparable genome sizes, and with more genomes available thanks to increased sequencing effort, we can therefore screen for a comparison of ERVs in this order. We are therefore interested in determining the comprehensive ERV content in Anuran genomes, and we're hoping that by looking beyond mammals, we can increase our understanding of ERV distribution and shed light on virus-host interactions and the possible role of ERVs in vertebrate evolution. Given the limited data on ERV content in Anurans, we aim to screen them by designing a bioinformatic pipeline in the study. And today I'll be talking about screening for alpha retroviruses. So this is an overview of the bioinformatic pipeline, and I'll just go through each of these steps now briefly. So we start the screening process with a T-blast for alpha retrovirus M protein, which is against the anuran genome assemblies. These sequences are filtered for quality, extracted, and aligned. And we can use these ENV alignments to find the 3' flanking LTR sequences. We can then use these LTR sequences in the next step of screening, where we use a BLAST-N for the LTR sequence against anuran genome. We then end up with 2 databases of LTR sequences and M sequences, which we can use to identify potential full-length insertions. And with that, we're looking for those LTRs which are less than 20,000 nucleotides apart with an M pit in between. And for this particular leg of the study, because we're focused on complete full-length insertions with the full genes in between the LTRs, we are looking for LTRs which have a high level of similarity, as these are identical when they're inserted into the genome. So we align our LTR pairs and look for those which have over 95% similarity. We can then extract from the 5' LTR to the 3' LTR and look for our internal genes and open reading frames. Once we found these, we aim to classify them using conserved domain and phylogenetic analysis. So in total, we screened 46 genomes, and this consisted of 31 frog species and 15 toad species. These ranged in genome size from 0.5 to 10 gigabytes, which are represented on this graph by these circles. From the initial T-blast-N for alpha retrovirus M protein, we found results in 36 of the 46 genomes. Of the 10 genomes which didn't have any results, 8 of these were contig or scaffold level assembly builds, and the rest of the contig scaffold level assembly builds are represented in this graph by asterisks. In the pipeline, we looked for solo LTRs, and these were identified in 35 of the 46 genomes. Solo LTRs represent when homologous recombination has occurred between the LTRs, resulting in just a single LTR. And the majority of these results, as you can see in the left-hand side of the graph, were in the Bufo species, as well as the Bombina species down here. And these numbers are similarly reflected for the potentially full-length elements. And again, these are the elements which have LTRs less than 20,000 nucleotides apart with an M in between and over 95% similarity of our LTRs. Next, we wanted to look for the complete full-length insertions, so those with GAG, POL, and M. And we identified these in 11 anuran species. We compared the sequences we found to viral databases and other studies and found 10 novel endogenous retroviral families. We've named them in this study as ANERV, so they're listed here from 1 to 10 for anuran ERVs. And you can see the species which they're present in, and in most cases, these are represented by multiple loci within the genome. You can see that there are 3 groups, ANERV2, ANERV4, and ANERV5, which are present in multiple species, as well as some species which have multiple groups in their genome, such as the Spear species and Bombina bombina. We next wanted to classify these 10 families using conserved domain and phylogenetic analysis. So here we have the POL protein phylogenetic tree and the M protein phylogenetic tree of our 10 insertions, along with representative retroviruses from each of the retroviral genre. It's apparent from our phylogenetic analysis that all 10 of our ERV families have a recombined form, and by that I mean their POL classifies with one retroviral genre and their M from the same family classifies with a different retroviral genre. We can see that the majority of our ANERVs which we identified in terms of the POL cluster with epsilon retrovirus. We can see that they're closest, most similar to other anuran ERVs which have been previously identified, and that they form separate group to the fish epsilon retroviruses here. In terms of our M, we can see that the majority of our anuran ERVs cluster with gamma retrovirus, and again are most similar to other anuran ERVs which have been previously identified. Epsilon retroviruses were not included in our M protein tree, as their structure is quite different compared to those that in beta retrovirus, delta retrovirus, gamma retrovirus, and alpha retrovirus. We were confident that none of our ANERVs had an epsilon retrovirus ENV classification due to the conserved domain analysis, as well as the protein alignment. We have two of our ANERVs which cluster down here with alpha retrovirus, and we can be quite confident of this because there is this distinctive M structure. And we can also see that, again, they're most closely related to other ANERVs which have been previously identified. And finally, we have two ANERVs in terms of the pole which cluster with this ERV-L group up here. So this is a summary of our classification based on the phylogeny for the proteins. We did actually undertake a GAG protein phylogeny as well, which wasn't shown in this presentation. However, this wasn't that useful in determining what genre our ANERVs cluster with, as there is so much variation in the GAG protein. So as I mentioned, the majority of our ANERVs cluster with epsilon retrovirus with a pole and have a gamma retrovirus classified M. Now, this structure has been seen previously in 4 other ANERVs, as well as 2 fish ERVs, and it has been hypothesized that by gaining this epsilon retrovirus, which gained a gamma retrovirus M, allowed the epsilon retrovirus to increase its host range, as epsilon retroviruses are typically identified in bird species. We have 2 ANERV groups which have this alpha retrovirus classified M. Now, alpha retroviruses are primarily identified in bird species and were proposed to be host specific to this group. However, this is based on the pole, and when we look beyond the pole for this alpha retrovirus M, we have seen that it's been found in other vertebrate groups, including a single anuran species, as well as just a solo gene in some reptile species. So although this alpha retrovirus group is proposed to be host restricted to birds, the alpha retrovirus M structure may be more common in other vertebrate orders, particularly in reptiles and amphibians, but it's gone unreported due to minimal research in amphibians and reptiles, but also with this focus on classification by the pole. Here we can see that our alpha retrovirus classified M has associated with an epsilon retrovirus in one case and this ERV-L in another case. So we have 2 ANERV families which cluster with ERV-L, and we can be confident of this due to the DUTPase, which is present in the 3' of our pole. Now, DUTPase is present in multiple genera of retroviruses, but in this specific location, it's only found in ERV-L. ERV-L is a group of ERVs which are found as ancient insertions in mammals, but they actually lacked this ENV gene. So in mammals, they're only found as GAG and pole. Since the discovery of these insertions in mammals, however, an ERV-L with this gamma retrovirus M structure has been previously identified in another anuran. So the ERV-L-like ANERVs, which were identified here, appear to have independently acquired an alpha retrovirus M and a gamma retrovirus M independently, and this again could possibly facilitate access to this new host species. So it's evident that recombination is present amongst anuran ERVs, and the resulting incongruences between pole and M highlight the importance of investigating and classifying our retroviruses based on more than just the pole gene. The recombination identified here, particularly involving the M gene, is not an uncommon observation, and it has been documented in ERVs, particularly in reptiles and amphibians, although it has been found across vertebrate species, and it's proposed to be a process of new ERV emergence and access to new host species. So to summarize, this study identified 10 novel endogenous retroviruses in 11 anuran species, represented by multiple loci in the genome. All 10 novel endogenous retroviruses showed a recombined genomic structure, which extends this pattern of recombination, which has been previously highlighted in 2 other anuran ERVs, as well as in amphibians as a whole. Alongside these complete insertions, we also identified multiple loci of partial ERVs and solo LTRs in 35 anuran genomes. With the bioinformatics screening pipeline which we designed in this study, we can continue to screen for novel ERVs in anuran genomes, aiming to achieve this comprehensive screening of ERVs. These initial steps in characterizing anagram herbs will provide a foundation for our understanding of the roles of herbs in the vertebrate order and add to our understanding of the complex evolutionary history of retroviruses in vertebrate evolution. If you're interested in this work, then please check out our publication on bioRxiv. And with that, thank you to my supervisors and my lab, as well as my funding, and thank you all for listening.
Hello, everyone. We have two of our speakers here today to take any questions that you have, and I think we have about 10 minutes. Ying issues her apologies. It's in the middle of the night in Canada, so she wasn't able to make it, but you can put questions on Slido or email her and she will address them later. So I guess the first question goes to Mickey from Rich Edwards, who says, Mickey, are frogs generally more susceptible to chytrid when stressed? Do you think there might be merit in selecting for frogs to be more tolerant of climate change to generally help them respond to challenges? Or do you think that maintaining genetic diversity is more important?
Yeah, thanks for the question, Rich. Well, a couple of questions, I guess. Frogs definitely are more susceptible, I guess, anytime they're more immune compromised, just like us. They have, you know, a complete immune system. So anytime you're going to stress frogs out, of course, they're going to become more susceptible to diseases, and chytrid is no exception to that. We didn't measure stress in our experiments here, but some people have done that and shown that obviously increasing stress can lower their immune system and make them more susceptible. So definitely something to think about when we're doing our experimental exposures. We do take some things into account to try and do that, like allowing the frogs to acclimate to their tanks when they're moved into the experiment. So they get 2 weeks in there. We minimize, we don't handle them unless we need to take samples from them. Then they've got places to hide in their tank and things like that. So we can try and minimize that for them. In terms of climate change, it's obviously another big factor. And the reason why frogs are threatened. For species like the crabberry frogs, we actually kind of predict that climate change might help them, given they're an alpine species. It actually, you know, kind of might warm up their environment and potentially create less refugia for the fungus. But for our tropical species, this is obviously a bit of a problem if they're already at their upper tolerance limit for temperature. And in terms of maintaining genetic diversity, this is crucial, of course, especially when we're talking about small populations and populations with small effective population sizes. With the breeding program that we're talking about, that optimized contribution approach does try to maintain that genetic diversity, as I showed, where we're actually, you know, increasing heterozygosity slightly, while also still increasing their genetic gain. So yeah, definitely something that is super important when we're talking about breeding animals. Thanks.
Thank you, Mickey. Okay, so now we have another question from Rich for Haley, who says, do you know how well the standard repeat annotation tools like LTR Harvest and Repeat Modeler are able at identifying herbs? Is your detailed approach just needed to get accurate models or counts, or do you think generic repeat identification will miss them completely?
Hi. Firstly, sorry for the terrible lighting. It's also about 5 a.m. here. So we did try using these tools to find herbs in anagram genomes, but because there's so little publicly available data which have been published on anagram endogenous retroviruses, these tools just weren't finding the same amount of detail that we can with this more detailed way. I think there's about 84 endogenous retroviruses which have been found in anagrams, and I think only 17 of those are in databases like NCBI. So these tools which use these databases to find them are just missing them.
Okay, great. We do have another question for you, Haley. And that is there any danger of contamination from retroviruses ending up in genome assemblies and giving false positive herbs? Or is there a robust way of telling them apart?
That's a good question. I use publicly available genomes, and I'm not really part of the genome assembly side of it, but I guess if it was in contact with the process of sequencing the genome, or the amphibian was infected, then it could be possible. But because they have these flanking long terminal repeat regions which occur once the virus is integrated into the genome, we can be fairly confident that they are endogenous retroviruses. And we can also be more confident when we find multiple insertions of these, so multiple occurrences of the same LTR sequences, and we can place these in different parts of the genome as well.
Very interesting. I have a question for Mickey, and just to remind everyone, you can put your questions in Slido. We have a few more minutes. So Mickey, doing a disease challenge study in an endangered species is quite unusual. Would you be willing to talk about how you had access to these animals and some of the challenges with doing all that work?
Yeah, it is. Thanks, Tiff. I guess with the corroboree frogs, we're in a bit of a unique situation with them. So because they are no longer in the wild, so they were the last remaining animals were collected from the wild and brought into captivity. And they've been breeding them in captivity now for nearly close to 20 years, and they've become really successful at, getting them to breed. So they actually, across the 3 zoos that breed this species of frogs, they produce anywhere from about 800 to 2,000 eggs each year, which they're releasing back into the wild, and they have been doing to try and, get them back out there. But unfortunately, because of the presence of the fungus, they aren't surviving. So for us, we were really lucky that due to the amount of animals they can breed, we were able to, get a large portions of those animals to use for our disease challenge. And I guess why we were allowed to do this, given they are critically endangered, is because these founding frogs are now getting quite old. So these animals that were brought into captivity, some of them are coming on 20 years old now and are potentially going to, stop breeding in the next couple of years. So they're really at the point with this species that if we don't try and do something different for them, they are just going to be maintained in this closed captive population where, yes, we're breeding them, and yes, we're trying to get them back out in the wild, but unless we come up with a solution, they are just going to be maintained in this captive population, which at some point will inevitably become inbred. So we were really lucky in some ways, unlucky in other ways because of the situation they're in, that, the zoos here in Australia are really willing and excited to try these different types of conservation methods that could potentially provide a solution to, reestablishing these frogs back in the wild. If that helps answer your question.
Yeah, thank you. That was great, Mickey. I have a question for you, Haley. Do you have any reason to believe that any of the herbs that you detected are application competent?
They definitely have the ability to replicate, as the 10 that we have identified all have complete genes. I can't tell at this current time, but it definitely would be an interesting thing to look into, especially because they have these LTRs which are highly similar. And because they're identical upon insertion, it shows that they are quite recent insertions. And so they could be replicating in the genome, and there could be either positive or negative effects of this.
Yeah, interesting. That will be something to stay tuned to with your research to see what you find. So very cool.
Thank you.
So we don't have any more questions in the Slido chat. If anyone has a real quick one they want to get in before we wrap up, that would be great. Okay. Doesn't look like we have any more questions. So thank you everyone for joining in to our symposium, and thank you to our 3 presenters, all 3 of which are PhD students. So it's great to see the amazing work that they're doing. And yeah, please feel free to reach out to any of us if you have any questions, either on Slido or via our emails. Thank you.